Hello everyone, here for the South American Microbial Research Network. I am with Stephanie Porter from Washington State University. Hello Stephanie, how are you doing?
Good, thank you for having me here.
So Stephanie, you recently published a really interesting review named Agriculture and the Disruption of Plant Microbial Symbiosis. Well, you talk a little bit about the influence of domestication on microbial communities below ground. So what can you tell us about this review? How did you have the idea of publishing a review about such an interesting topic?
Well, the co-author Joel Sachs and I have been collaborators for a long time, so we've discussed ideas back and forth, and both of us have focused on other research, but both of us realized our interests were converging on, what are the differences between how crops interact with soil microbes and how wild plants interact with soil microbes? Because both of us have studied a lot of wild plant interactions with microbes and are now focusing more on the impacts of agriculture on the soil microbiome. And so we wanted to contrast these kinds of scenarios to see how they differ. Crops are dramatically different from wild species in terms of the environments we create for them, how the things we want them to do, the breeding that we've exerted for thousands of years. So we just think there's a lot of differences, and we want to understand them systematically. So when people started domesticating crops thousands of years ago, what has happened to these plants' interactions with their microbes over those thousands of years of evolution? And in particular, more intensive breeding and more intensive agriculture in the last century or a half of a century have also had, major impacts on the way agriculture happens, and the germplasm that exists today in high-intensity agriculture is very narrow in terms of genetic diversity and very specialized in terms of the things we've bred for. But we know so little about what's happening underground. What are these plants still as cooperative with microbes? Are microbes still as important for them as they are for wild plants? Or have we bred plants to be more independent of the soil microbiome and rely instead on intensive agricultural methods? So these are some open questions that a lot of people are trying to answer. And so we wanted to bring together these diverse ideas into a paper that summarized where we are right now with our understanding, and to lay out some questions to help the field move forward, integrating these evolutionary concepts with the study of plant microbe interactions in agriculture.
Okay, and so in your review, it seems that in most of the studies that have been done, in general, domestication has not been very positive, right, for microbial communities.
Well, on that point, that the question is open.
Okay.
We summarize some literature that shows reductions in the diversity of symbionts and in the benefits symbionts offer to plants and in the ability of crops to control their symbionts. But we didn't conduct a meta-analysis, which would be a systematic review of what the literature says. Okay. So we're really looking for examples. And there may not be enough literature at this point to do a robust meta-analysis. So I think it's really important. We wanted to encourage more people to address this question. And we use the word disruption in the title because there's a lot of ways that agriculture reduces the interaction between plants and the microbes. Because, for example, the mycorrhizae may be less abundant in agriculture than they are in a natural system. But we don't know if maybe it's helpful for crops to interact less with soil microbes. Maybe it's harmful. We don't really know. There's not enough data. So I just wanted to put that out there, that I think that's a frontier still.
Okay, but what is your feeling about this? Do you think that, well, especially intensive agriculture has been negative for the microbial communities, and do you think this is a problem?
Well, certainly that's what my lab is studying right now. It's the question I'm really interested in. And a lot of our work is focused on the legumes and rhizobium interaction in agriculture. So, for example, my graduate student, Niall Millar, he has tested 5 different domestication events in the legumes and compared the benefit that plants get from rhizobia for crops and their wild relatives. And he finds that in 2 cases of legume domestication, the crop germplasm gets less benefit from rhizobia than their wild relatives. And in the paper, we summarize these kinds of tests that people have done. You know, the Zhang et al. Paper from 2019 suggests that plants developed after 1950 benefit less from inoculation with mycorrhizal fungi than plant lineages developed for agriculture before 1950. So there are pieces of evidence like these. Whether that's the whole story, I don't know, because we don't have enough data, but certainly it can be detrimental in terms of the intensive breeding that we've done can be detrimental to the symbiotic function of crop plants.
Okay. Yeah, you talk a little bit in the paper. Also, it depends on the type of agriculture that you want to develop, right? So maybe that's for intensive agriculture. It's not going to be detrimental for the plants, but then if these plants are highly selected, and then you go back to more sustainable agriculture in the term of you don't use so much fertilizers or herbicides, then maybe the plant would not be so well adapted to that kind of agriculture, and it would be difficult for the plant to kind of go back to less intensive agriculture.
Right, and this is where I think more research on diverse crop germplasm would be useful. So, you know, we don't really know what sets of crop germplasm are optimal for obtaining the most benefits from symbiosis with soil microbes, because in many cases that hasn't been tested. But identifying lineages of crops that do perform well under different kind of agricultural scenarios would be the next step probably to trying that, and to see if you could breed for crops that are thriving under alternative agricultural practices and maybe benefiting more from microbes instead of from more intensive inputs.
So is it some kind of the research that you're doing in your lab? How would you select for these crops? Like, I mean, you mean in the genomes, looking for specific genes responsible for interactions, or how would you do this?
Well, with the legumes right now, we have a new project that was funded to investigate the effects of domestication in the legumes on the rhizobial symbiosis. And so we are working with the USDA germplasm repository, GRIN, and testing many lineages of legume crops from dozens of domestication events in the legumes. So comparing wilds to land races to modern cultivars and breaking down in detail the interactions they have with rhizobia, how much they benefit from them under high and low fertilization inputs in agriculture. And so it's a big job, and it's going to take 5 years. But thematically, we're going through a lot of these because we want to characterize the differences between all these different classes of lineages so we can understand, well, what happened? And this would also allow us to identify those lineages that perform well under low fertilizer inputs and get a lot of benefit from the rhizobial symbiosis. So that's one case study, but I think that kind of approach could be done more broadly. We have these wonderful germplasm repositories that have everything from the wilds to the ancient land races to modern cultivars available for researchers, and it's a great tool for investigating these questions.
You talk a little bit in the review about the accumulation of deleterious alleles, right, that are a consequence of domestication. And it's actually a way of looking at domestication, past domestication events in the genomes. So is it something also that you're working on, or you think that could be done with microbial research, for example?
So I think that there's a lot of potential to combine our understanding of the accumulation of deleterious mutations due to domestication. So we know that when we domesticate organisms, we reduce their population sizes, their effective population sizes. We inbreed them, we accumulate a lot more deleterious mutations in the genome that go to fixation in our crops. And we have a lot of evidence that these deleterious mutations can have negative consequences for crop function. But we don't know as much about how that process may impact symbiotic function. So one of the ideas we're exploring is in the genes that we do know are involved in symbiotic function, how abundant are deleterious mutations that are more abundant in domesticated lineages than in wild lineages. So I have a grad student who's working on that question for the legumes right now. Another way we're trying to approach this is by using recombinant inbred lines. So for a lot of our major crops, we have crosses that have already been done between wild and domesticated lineages. And so and then those crosses have been propagated for, 6 generations of inbreeding to generate recombinant inbred lines, which are mostly homozygous through their genome, but have chunks of the genome that are either wild or domesticated alleles from the original wide cross. And these allow us to, we have hundreds of these lines. We can measure their symbiotic function and then map these symbiosis traits to different regions of the genome and either the domesticated or wild allele having a positive or negative impact on symbiotic function. So that will help us localize quantitative trait loci that are important for symbiotic traits that we care about. So this is another way we're trying to approach identifying potential genetic drivers of the evolution of symbiotic function during domestication.
So in general, would you think that increasing the genetic diversity of the plant is one way of maybe increasing the diversity of microbes that they're able to associate with?
Well, there's a lot of debate about the relationship between microbial diversity and ecosystem function, right? So this is not a simple relationship. And so tracking microbial diversity is not an easy way to track the function that you're interested in observing in a soil. In this paper, we focused a lot on crop genetic diversity or plant genetic diversity because we think that there's a lot of reason to explore variation in how plant lineages control their symbionts and are able to select among their symbionts. And the plant-focused side of the control on microbial diversity, rather than sort of the bottom-up side of looking at microbial diversity and the impacts on plants. Because we know there's genetic variation among plants for the diversity of symbionts they associate with. There's genetic diversity among plants for their ability to choose amongst partners that differ in quality in the soil. And this means that plants will evolve, and we can evolve plants too to accentuate the traits that we care about. When we use microbial inocula in the field, we know that there's a lot of issues with maintaining a long-term effect of inocula that we may want.
Exactly.
One inoculum can have very different effects in 2 different fields because the environmental context is very important. So we suggest that it may be possible to have a more lasting and stronger impact by modifying plant germplasm to affect the microbial communities we want to see, or that will in fact benefit the plant more than by trying to manipulate the soil microbiome directly. That's one of the ideas we propose. And I think what we need now is a lot of studies to test that. Is this a more fruitful way forward?
Yeah, exactly. I agree. And I hope that the people who are listening to us will agree too and start do more research about the subject. So thank you very much, Stephanie. Do you want to tell us a little bit more about you have other projects you're interested in?
We have a couple projects in the lab. One is looking at how these rhizobial bacteria adapt to heavy metal stress in the soil. And the other is looking at the effect of domestication of legume crops on plant microbe interactions. So probably your listeners are most interested in that second project. And right, we're taking combined approaches from looking at genomics for existing genomes that have already been sequenced in the legumes. There's a lot of existing data. We're using field trials of the recombinant inbred lines and the wide samplings of diversity and measuring nitrogen fixation in the field to see how well symbiosis supports plant health in actual crop germplasm. And we are also doing greenhouse tests to modify rhizobia to be more or less cooperative, and then measuring very carefully in the greenhouse how well domesticated versus wild relatives in the legume crops perform at excluding those bad symbionts that are not very robust nitrogen fixers. So we're really dissecting it in more detail there. So those are some of the big foci for the next 5 years in the lab.
Okay, really cool. Yeah, so you're doing a lot of experiment too. That's very exciting.
Yeah. I mean, it's going to be a long haul, and certainly the pandemic has slowed down our ability to do research, but we'll get back up online once, things have returned to normal.
Yeah, exactly. Well, thank you again for your time, and congratulations on this very interesting paper. And we will be back soon for more interviews for the South American Mycorrhizal Research Network. Bye.
Goodbye. Thank you.